So in lecture, we're starting to get into some important things about DNA and RNA. So right now these things are going to be pretty simple, but it's important that you understand them so that you're not lost later. So let's start with this question. You are given the following DNA sequence. What is the sequence of its complementary strand? So one tip to help you always, anytime you're dealing with DNA, RNA, throughout the rest of this class, when you're given a strand like this, always start by labeling the ends. So one end is always 5' and the other is 3'. So this is our leading strand here. So it's going to go 5' to 3', and then our complementary strand is going to be the opposite. It's going to go 3' to 5'. So that's just a good tip for the first thing to do. So next, let's look at the actual content of the question. What is the sequence of the complementary strand? So in order to figure that out, complementary strand is going to be the opposite. So T's will become A's and vice versa, and G's will become C's and vice versa. So go ahead and pause the video, write out this DNA strand, 5' to 3', and then write its complementary right underneath it. Pause the video, take a few minutes to do that, and then I'll pop the right answer up. So this is what you should have gotten here. So like I said, as you go along, it's going to be the opposite if you use this key that we've got here. And so then this sequence runs 3' to 5'. So our next question to consider involves translation. So the next question, how many amino acids would there be? How many amino acids long would the chain be after translation? So if you remember, amino acids are made up of 3 nucleotide bases. So the first one here would be ATG, and the next GAC. So the way to figure this out is you can either count up the total number of nucleotides and divide it by 3, or you can just draw little lines in groups of 3s and count how many groups you have. Go ahead and take a minute on your paper that you wrote it out on, do it by yourself, and then we'll do it together. So as you can see now on your screen, I've split this chain up into different groups of 3. So the reason I do it this way is because in most questions you'll be asked to figure out which amino acid each codes for. And so when you have them in groups of 3 this way, it's easy to reference your chart and figure it out. For this question, we didn't necessarily have to break it up this way, but I like to do it this way. So now we can count and we can figure out how many amino acids this would be. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 sets of 3. So our amino acid chain is 10 amino acids long. Don't forget that this last amino acid is the stop codon, the TAG, or there's 3 different ones, but it doesn't actually code for an acid, it just tells it to stop. And so it's 1 less than that, so our answer would be 10 amino acids long. Another important thing to remember along with recognizing that stop codon is that you always have to recognize the start codon. So in this case, let me get a better color there. In this case it's ATG, or as you'll see it on the table, AUG. So you may be given a problem with a long DNA sequence and the start codon may not be the very first one that you have. You may have a couple of bases before that, so you need to scan through and look for that ATG, and then you say, okay, well this is where the actual gene coding section of it starts. So then you start there and then you start translation. So always be careful to watch for that, because you will see that come up in problems as you do homework and as you do tests.